Strawberries grown on innovative substrates like compost, biochar, chitin, or wood fibers grow just as well as they do on traditional substrates, which contain a lot of peat. And that is big news. This means that in the relatively short term, the sector can become even more climate-friendly and even more circular than it already is. The greenhouse sector has already been looking for alternatives to peat. During peat mining, massive amounts of CO2 are released into the atmosphere, warming our planet even more.
Wordt heel veel veen gebruikt in teeltsubstraten, maar veen komt natuurlijk uit veengebieden en dat zijn belangrijke koolstofreservoirs. Dus er is heel veel maatschappelijke discussie of het wel zinvol is om die veen te ontginnen voor gebruik in potgrond.
A good technical replacement is coconut shells, but those raise some serious sustainability questions as well. Besides, the specialists expect that peat and coconut will become more scarce and thus more expensive.
Kokos is een schaarste aan de gang, alternatieven zijn nodig en we hebben hier nu een proef liggen waarbij dat we alternatieven gebruiken en ook succesvol gebruiken.
Deze substraten zijn eigenlijk vernieuwend omwille van dat een veengehalte wat lager ligt. Dus dat is ook een vraag vanuit de sector. Dus dat veen dat ligt echt onder druk.
In the European Horti-BlueC project, the Belgian research centers Ilvo, Hoogstraten and other partners have done extensive trials to make potting soil with a number of local alternatives, all of which are raw materials that are widely available and can easily be mixed into potting soil. Some lesser-known products showed promise as well, such as biochar and chitin.
Chitin is eigenlijk een chemische stof die planten meer weerbaarheid kan geven tegen ziektes, maar die ook een bron is van minerale stikstof, dus stikstof die de plant nodig heeft om te groeien. En dan hebben we biochar. Biochar is eigenlijk zeer stabiele koolstof die bij hoge temperatuur geproduceerd is. We hebben ook biochar op basis van vlaslemen, dus ook een bijproduct van vlasteelt.
At least 40% of the peat can be replaced with no effect on yield. In terms of disease susceptibility, fruit quality, and shelf life, the strawberries grown on substrates without a lot of peat scored just as well as those with a lot of peat.
Uit alle voorgaande proeven en ook deze die dan nu lopen is, zien we eigenlijk geen verschil tussen de substraten dat we aan het testen zijn.
The new substrates can get a second life at the end of the growing season. The researchers could make them into compost and could even convert them into biochar, and then they were mixed into new substrates. The used substrates enriched with biochar were also good soil improvers, with a special climate benefit. They store additional carbon in the soil.
Dus bijvoorbeeld op basis van gebruikte potgrond kunnen we ook terug biochar maken die we dan voor diverse toepassingen kunnen gebruiken. In potgrond, maar die compost, die gebruikte potgrond en die biochar, die kunnen we ook gebruiken om koolstof op te slaan in landbouwbodems, om ons eigenlijk te wapenen tegen de verdere klimaatverandering. En dat kan ook een extra inkomensbron zijn voor de telers die gebruikte substraten afleveren.
Reusable, local, effective, and good for the climate. These alternative substrate recipes are very promising. We'll be using more substrates in the future for various reasons. First, growing on substrate has serious advantages. It uses less water for the same yield, has better disease management, and the plants are less vulnerable to extreme weather conditions. Secondly, the demand for substrates is growing because cities are becoming greener. Conclusion? These innovative substrates make the grower's dream come true. A big jump forward in sustainability in a perfectly feasible way.
Telers reageren over het merendeel positief over de experimenten die hier nu lopen. Het belangrijkste voor hen is natuurlijk dat er niks verandert. Dus dat de omzetten blijven behaald worden, dat de opbrengst hetzelfde blijft, dat de kwaliteit van de aardbeien niet in gedrang komt. Maar dat is het geval. En dat maakt voor telers de stap nu wel gemakkelijk om die omschakeling te gaan maken.
Want to know more? Contact us today.
Good afternoon, everyone, and welcome to the fourth webinar already from Horti-BlueC. Today we're going to focus on the practical trials that we've done with the new growing media blends that you have just seen in the video. So actually the video was a perfect introduction to this webinar. My name is Peter Meelis from Research Center in Hoogstraten, Belgium. I'm a researcher in strawberry, as you have seen as well in the video. I'll be your chair today, so I'll be collecting all of your questions. I have to say that to ask questions, we're not going to use any hands we're not going to enable you to speak and ask your question, but you can put your questions into the chat. So please use the chat if you have any questions. We will answer them at the end of this webinar. I will monitor all the questions and I will point them toward the right speaker to which the question is addressed. So I will take care of that. I will also introduce all the different speakers, as you can see with the background of a sea from Horti-BlueC. So that's really obvious now. I also have to mention that this webinar will be recorded. So if you don't want to be in screen, please turn off your camera. The questions at the end of this webinar will not be included into this recording that will be put on YouTube, but all the rest of it will be put on YouTube, so you can find it there afterwards. Also, to allow a clear talk for each presenter, I would ask everyone to stay muted during the entire webinar. Your questions will be answered by the different speakers and me. Some of the speakers have prepared some polls where you can answer questions. Those polls will be anonymous, so your name will not be recorded with your answer. We can only see the amount of answers, but not which persons have answered them. Today's agenda, you can see here, we have the different speakers that we'll go through and they will talk about their own expertise. I'm not going to go over all of this because time is quite limited. So I'm going to go towards the first speaker. That's Bert van de Kastelen. He's from Ilvo, and he is actually the promoter of the project that we're working in all together. That's called Horti-BlueC, and he will give an introduction about the valorization chains that we did and used during Horti-BlueC. So Bert, the word is yours.
Thank you, Peter, for the introduction. So let's get started with this fourth webinar. So we will present results of the Interreg 2Cs project Horti-BlueC. The project has been executed by 9 partners and it has also been co-financed by 3 provinces. So in this project, we have some new solutions that we want to showcase. So we focused on the reuse of spent growing media. We did work on new additives like biochar and chitin. We have tested new blends for strawberry and tomato cultivation, so you will see these results today. And we also focused on new technology, so biochar production, gasification for biochar, but also for heat and for energy. So we started from 5 residual materials that we have upcycled during the project. And for doing that, we combined a residual material with a certain treatment process. And based on that, we have produced building blocks for sustainable growing media. So this was quite an issue to find the best technique to process a high quality building block for growing media. But based on our expertise, we were able to define 4 valorization chains during the project. And the first valorization chain, it was on the use of biochar and the production of biochar. The second valorization chain, it was on the production of chitin. The third valorization chain, it was on spent growing media and it's upcycling. And so now we end up with the fourth valorization chain. It is on bulk replacement in growing media. So these 4 valorization chains were the topic of 4 different webinars. As you can see here, so today we have the last webinar. And if you want to know more about the previous webinars, you can go to the website, but also to YouTube, where you can find the complete recording of each of the webinars and also the presentations and the fact sheets. But now the topic of today. So we will talk about our dream, because nowadays growers have to buy growing media to grow tomatoes or strawberries. They produce high quality fruits and vegetables, which they can sell for a good price. But then at the end, they sometimes end up with spent growing media they have to get rid of and they have to pay for that. So it's really a waste material. So our dream is actually to start from another blend, a sustainable blend, which also allows to grow healthy strawberries and tomatoes. And at the end, we have a spent growing medium that can be easily upcycled. So maybe it can also be an additional source of income for the growers. So this upcycled spent growing media can be used in different ways. And this solution, it's good for the planet as well. So it's our dream that we will explain today. So the two crops we have looked at are tomatoes and strawberries. And for tomatoes, we will showcase a 100% organic mix that can be used as replacement of Rockwool or coir. So these are actually the references in the project area. And on the other hand, we also have results for strawberry blends, where we have peat reduction by 40%. So these results we can compare with peat blends or with coir blends as well. So these blends for strawberry, so here you can see that we have replaced peat and coir by wood fiber, green compost in these blends. While in the blend for tomato, we have also used wood fiber, bark, green compost, and also coir. But in the future, it maybe is possible to even substitute also the coir in the blend. Beside this bulk replacement, we also have used two products in very small amounts. So it's chitin and biochar, so the salt and pepper of the growing media. So we have used it at very small amounts of 2 grams per liter. And in some cases, we also did some bulk replacement by biochar. So in higher percentages, we have also used this biochar in these blends. So this brings us to the topic of today, because the current blends that are used, like peat, coir, or mineral wool, they mainly focus on one function. It means to give structure to plant roots. So it's very important for them that you can have good plant production and a good yield. However, we feel that by using new materials, we can also reach a certain degree of ecological intensification of growing media and soilless cultivation, because we can also more use the microbiological properties of these blends, and we maybe can also use nutrients in a more efficient way. So, for instance, we will talk about compost. Compost can have different functions in growing media, but we will also focus on plant fibers. And these plant fibers can also have different functions. And it's a quite big group of materials, because it contains like wood fibers, but also the woody fraction of green compost. It can also contain like plant fibers like reed or other materials that we can harvest in nature conservation. So during the management of these areas. It can be crops like miscanthus, which we grow, especially for having these plant fibers, or it can be like crop residues, like flax chives that we have as a crop residue available. So when we talk about the microbiome of growing media, we can have different materials, as said before, but we can have also different characteristics that we look at. For instance, we can focus on the microbial biomass, which is available in the blend or on the materials, but we can also look at the microbial activity, which is mostly related to carbon mineralization, but also to nitrogen mineralization or immobilization. We can look to the diversity of the microbiome, so the number of species which are available. And we can also look to specific microbial functions of this growing media. So in the agenda, you also saw that we promised to present results for the LCA study on new growing media blends, but unfortunately we had to decide to postpone it to the beginning of 2022 because our results are not completely finished yet, but you will hear again from us next year, and we will provide you with the full results of the ongoing LCA study. So thank you for your interest in this webinar, and I think we can now really start with the webinar.
Thank you, Bart, for your introduction, and for also mentioning already the different blends that were used in this project, and that will be introduced and presented in this in the rest of this webinar. One of the partners within Horti-BlueC was, of course, a substrate provider and a distributor and producer. It's one of the major ones in Belgium. It's the company called Aharis. And one of their experts is present here, and he will give a next presentation. It is Jeroen de Zaytijd. He's innovation manager at Aharis, and he will talk about the pros and cons of the new different blends that we've used within Horti-BlueC. So, Jeroen.
Yes, hello, good afternoon, everybody. Thank you, Peter, for the introduction. So my name is Jeroen de Zaytijd. I'm innovation manager at Aharis, a company that produces growing media for the professional sector, but the hobby market as well. We are partner in the Horti-BlueC project, and we were tasked with the production of the experimental substrate blends that were used during the different growing trials. And today I will tell you something more about the pros and the cons of these experimental tomato and strawberry blends. So first I would like to tell you something more about the properties of a good growing media. And since these characteristics are dependent on the raw material constituents of the substrate, I will also talk about the composition of currently used growing media, but also about the pros and cons of alternative raw materials. And I will finalize by summarizing the pros and the cons of the alternative Horti-BlueC substrates that were tested. But first things first, so what makes up a good growing media? Well, we can identify three driving factors, being performance, but also the economics of a substrate and also the environmental aspects. So talking about performance, so of course the main purpose of a good substrate is to give plants an optimal environment for their growth and development. And a good substrate provides the plants with physical support, but also water, air, and nutrients. And the performance of a substrate is determined by its physical, chemical, and biological properties. Some examples of physical properties are the water-air ratio, the water holding capacity, the porosity, and the bulk density. An ideal substrate, for example, is not too heavy and balances the water and air ratio content to the needs of the plant. And this in turn is dependent on porosity and size distribution. Next to the physical properties, also the chemical properties are equally important. So plants prefer a pH of 5 to 6.5. They also don't like to be in very salty environments. And preferably the substrate also has a cation exchange complex, which means that it can buffer and hold on to nutrients. And next to the physical and chemical properties, also the biological properties of a substrate are very important. Of course, you want the growing medium to be free of pathogens, pests, weeds, and other residues, but you also want it to be biologically stable. This means that the carbon source inside of the substrate will not be decomposed very easily by microorganisms, because this goes hand in hand with nitrogen immobilization. But Bart will tell you something more about this later in this webinar. And next to performance, also economics are very important for a substrate. Think about accessibility and availability of raw materials, but also processing and transport costs. And increasingly important is the environmental aspect. So a good substrate should be sustainable as well. And it is very important to know that properties of a growing medium are determined by the properties of its constituents. So during the Horti-BlueC project, we focused on increasing the sustainability of substrates used in the strawberry and tomato production. In case of the strawberry production, peat-based substrates are today the predominant type of growing medium used. And also a lot of other plant production systems are still heavily peat-based. So that's why I will tell you first something about peat. On the other hand, tomato production, there the most commonly used substrate today is Rockwool mineral wool. So I would like first to highlight these two raw materials. So let's start with peat. From a performance point of view, peat is actually an ideal raw material for growing medium. This is also the reason why peat is still the most important raw material used for growing media today. Peat has a very good water availability and air content, also a very high water buffering capacity, has a low bulk density, so transport costs, for example, are low. Peat also has a very low pH, and this is very convenient because the pH can easily be increased to the desired level by adding lime. The opposite, however, would be very difficult, decreasing the pH of a raw material. And next to a low pH, it also characterized by very low salt levels. So you could also increase the nutrient levels to the desired levels by using fertilizers. Next to this, peat also has a cation exchange complex, so it can buffer nutrients. It's biologically stable and mostly free of weeds and pathogens. Not only from a performance point of view, but also the economics of peat are quite good. So it's good available and has a low processing and transport cost. However, from an environmental point of view, there are some downsides. So peat is considered non-renewable material. And it is known that the harvest of peat is associated with CO2 emissions. However, I would like to take this opportunity to emphasize that in contrast to popular belief, most of the peat is not harvested from pristine peat bogs. For example, our peat is RPP certified. This means responsibly produced peat. That means we don't harvest peat from pristine peat bogs, but from previously drained peat bogs that were drained earlier, for example, for agricultural land use or forestry. And after harvest, we also restore the peatlands with natural state. So that was peat. Tomato production is predominantly done on Rockwool. Rockwool also has a good performance. It has a high water availability and air content, also low bulk density and low EC. It's also biologically stable and free of weeds and pathogens. However, the water buffering capacity is somewhat lower than that of peat, and it doesn't have a CEC complex. Also, because it's a very sterile and inert material, there is no microbial life inside, so it could be potentially colonized easier by undesirable microorganisms. From an economic standpoint, production is sometimes energy demanding, and also recycling costs are quite high. And from an environmental standpoint, it's also a non-renewable material. So because of these environmental concerns associated with peat and rockwool, during the Horti-BlueC, we investigated whether more sustainable growing media could be produced and if they could perform as well as the traditional substrates. So for the strawberry trials, we replaced 75% peat-based reference substrate by an experimental substrate containing only 45 peat. And this peat reduction was accomplished by using raw materials such as wood fiber, green compost, and biochar. For the tomato trials, we replaced rockwool by a substrate that was for 100% made up of renewable organic raw materials like cocoa fiber, bark, wood fiber, green compost, and also biochar. So as mentioned before, the properties of a growing medium are determined by the properties of its constituents. So that's why I would like to dive into the pros and cons of some of these alternative materials. So let's start with green compost. Green compost has a good water retention. If it's mature compost, it's also biologically stable and free of weeds and pathogens. And it even has some pathogen suppressive properties. On the other hand, air content is lower. It has a heavy material, so a high bulk density. It also has a high pH, a high salt content, and only medium cation exchange complex. With regards to economics, it's a rather cheap material. It has a low processing costs, but because of it's a heavy material, transport costs are a bit higher. And if this material will be increasingly used in the future, there might be some concerns with regard to availability of high-end compost. The good thing is, of course, that it's a renewable material. Wood fiber is also an alternative material that is often used as one of the peat replacements. It has a high air volume, a low bulk density, good pH, and low EC. And because of high temperatures during production, it's also free of weeds and pathogens. However, water buffering capacity is low. There's no or only minor CEC complex, and it is potentially biologically unstable. That means that the carbon could be used by microorganisms, and there's a risk for nitrogen immobilization. With regard to economics, for now there's a good availability, but as you all know, wood is used in a lot of sectors, a lot of markets. So in the future, there might be some questions with regard to availability and rising prices. And also production is quite energy demanding. However, a good sustainability factor is that it's of course a renewable material. Then composted bark, when mixed in a substrate, it can increase air content of the substrate. It has an acceptable pH and acceptable EC. And if composted, it is also biologically stable and free of weeds and pathogens. However, water retention is lower. It's a heavier material and only has a minor CEC complex. The economical factors are similar to wood fiber because, of course, it's a side stream of the wood industry. Good thing is also here it's a renewable material. Then finally, also cocoa products are often used. These have high water availability and air volume, also good water buffering capacity and acceptable bulk density, also acceptable pH. They are biologically stable and mostly free of weeds and pathogens. However, a big downside of cocoa products is the high salinity level that they possess. And economically speaking, there are increasingly concerns about availability, and they are also characterized by high transport costs. These materials come from the far east, so from Asia, so that's a concern. And environmentally speaking, it's a renewable raw material, but also a lot of fresh water is needed to rinse out all the salts in these products. As mentioned, in some cases, we also used biochar as a bulk replacement in the growing media. However, I will not go into too much details because this was the subject of webinar 1. If you could not be there during the webinar, you could still find the video on the website and on YouTube. But biochar can increase air content, has a low bulk density, is biologically very stable, and could be a habitat for beneficial microorganisms. However, sometimes has a high pH and high EC depending on the input material. There are some economical questions still. Costs and prices today are quite high, but maybe there are some possibilities on a local scale where a grower produces biochar and uses the heat that comes free in the process to heat the greenhouse, for example. But a very good side of biochar is that it's like a carbon negative raw material because during pyrolysis process, the carbon is very stable, stably sequestered in the material. So to summarize, I will highlight the pros and the cons of the alternative growing media used during the Horti-BlueC project. So these consisted of these alternative raw materials. And we will compare the pros and the cons to the baseline scenario, which is a Rockwool for tomato production or peat used for strawberry production. So some of the advantages is that we use local, circular, and organic materials. So that's from a sustainability standpoint, very good. And this also allows for reuse and recycling of substrates. For example, biochar could be made from the used substrates or composts. Also, production can be less energy demanding than, for example, production of rockwool. And since these are organic and natural raw materials, they already have microbial biomass, so they are less prone to colonization by harmful microorganisms, for example. And on the other hand, it might be easier to colonize them with beneficial microorganisms. And what was, of course, very good is that in all these trials, we saw a comparable yield without making any adjustments in irrigation and fertigation compared to the reference substrates. Some downsides might be, what is still a question mark is the willingness of growers to switch to these new substrates. Also, the characteristics of some of these raw materials like green composts can vary slightly depending on the batch. As mentioned, there might be some question marks regarding availability and costs of these alternative raw materials in the future, so that has to be seen. Other small remarks are that, for example, the slabs of these alternative materials were a bit heavier than, for example, Rockwool, so it's more labor intensive. And also because we used green compost, there were some little flies present, for example. And then this brings me to my last slide. So some take-home messages that I would like you to remember from this talk. So a good growing medium is defined by its performance, economics, and sustainability. And all raw materials have their pros and cons. But there is a very positive trend going on towards the development of more sustainable growing media. Sustainability is increasingly important, and this is evidenced by, for example, the Horti-BlueC project, by joint efforts of research institutes, but also growing media producers and end users to all go into the direction of more sustainable growing practices. Thank you all for your attention.
Very good, Jeroen. Nice presentation. Also, thank you for providing all of the rest of the partners with the required blends that we could test in tomato and strawberry. I would like to remind the participants of this webinar that the chat is open. So if you have any questions, please put them into the chat so we can give them towards the right speaker at the end of this webinar. To switch over, we're going to see the first results of Horti-BlueC that we've did with the different blends. I'm going to give the word back to Bart van de Kasteel for his second talk. Bart is the senior researcher at Ilvo in Merelbeke, Belgium, and he will talk about the microbial activity in growing media, and he's highlighting the interaction with nitrogen. So Bart, please go ahead.
Okay, thank you, Peter. And I have already a question for the audience. So there is a first poll, and it's on stability. So we want to know what you think about stability. So what is the relationship between stability, microbial activity, and CO2 production? So you can answer the question now. And later on in the presentation, you will see the answer, of course. So this presentation is actually maybe a fairy tale. It's about the three little pigs. So they all had a different house, and depending on the house, it defines also the microbial activity. So you have the pig with the stone house, and it's very stable, but there is no microbial activity. So it means that there is no risk for a low stability, but there is also no risk of, for instance, nitrogen immobilization, but also no beneficial effects of microbiology. In that case, on the other hand, you may have the straw-like materials, which are very high in organic matter. They can immobilize nitrogen, which is not good, of course, but you can also have some beneficial microbial activity, maybe. So when we talk about the microbiome of growing media, sometimes we have different expectations. So actually, we expect a high stability, and as you have probably seen in the poll, it means, of course, a low microbial decomposition. So we see that a high stability means low microbial activity and a low CO2 production. So thank you for giving the good answer. So on the other hand, we also expect maybe a high microbial biomass, a high microbial diversity. We expect a low risk for pathogens or diseases. We also don't want nutrients to be immobilized or that there is retention of nutrients. On the other hand, we hope that the nutrient release and the availability for plant roots is optimal. And we also want optimal microbial functions, for instance, nitrification, so the conversion of ammonium to nitrate, nitrogen, which is more beneficial for plants, plant roots. However, these different expectations we have related to the microbiome of growing media, it might sometimes give some contradictory effects. So first focus on the stability. And when we want to assess the stability of growing media or the materials that we use for it, we can look at the CO2 emission, but we also can look at oxygen consumption. For instance, when we focus on CO2 emission, we have a very simple system tested in the project. So we filled a ring with 2 liters of materials, and we just measured the CO2, which was emitted by this mixture when it was mixed with chemical fertilizer to avoid any nutrient deficiency. And of course, the higher the CO2 emission is, the lower is the degree of stability. On the other hand, we can do the same with the oxytop method, where you do not measure the CO2 emission, but you measure the oxygen which is consumed during decomposition. So here also a higher oxygen consumption means a less stable material. So I can present you now results for different materials, different categories of materials. So we have different samples of each material that were analyzed. And when we focus on the chemical properties, we see that most materials are very high in organic matter content. The only exception is sometimes green compost, which has a lower organic matter content. And this green compost is then also characterized by a very low CN ratio. So it means it has a higher possibility for nitrogen release than the other materials. And we also see differences between the materials, for instance, in pH. So when we then look at more microbial related properties, for instance, if we look at nitrogen immobilization risk, we see that mainly the last group, so the soft plant fibers, they have a high risk for nitrogen retention. And then when we measure the total microbial biomass in the organic and the material, we see that peat and wood fiber, they are quite low in microbial biomass, while other materials, they are quite high in microbial biomass. So here it's very important that you express the microbial biomass on an organic matter base, because that's the source where microbial biomass can live actually. And then we have the 2 stability indicators. So we have both the CO2 release and the oxygen consumption. And also here, it's very important to point at the fact that it's expressed on an organic matter base. You can also do it on a volume base, but please never do it on a dry matter base, because these materials are characterized by differences in dry bulk densities. And if you express it on a dry matter base, you can have very strange results actually. So we see that materials with a higher microbial biomass, they also have a lower stability. The only exception is actually the plant fibers, where we measure a very high CO2 release, but the oxygen uptake rate is quite low for these materials. So there seems to be a discrepancy, and that's currently a work in progress that we are focusing on. But I think in general, we can conclude that the new materials that we use, they have more microbiology, but we have to figure out whether it is positive or negative for the blend. So besides the materials that we focused on, we of course also have mineral materials like mineral wool or perlite or vermiculite. On the other hand, we have biochar, which is very stable carbon. And when we compare them with the organic materials, it should be clear, of course, that mineral materials, they don't contain any carbon, of course, and they have a very low microbial activity and also a very low microbial biomass. In contrast, biochar is very rich in carbon, although this carbon is very stable. So you also have a low microbial activity and also a low microbial biomass. So in comparison with these 2 materials, we see that the other organic growing media constituents, they are much more important in terms of microbial activity. So then we come to the fertilization or the fertilizer application. So when you use chemical fertilizers or fertigation, your mineral nitrogen is directly available to plants. However, when you use the organic fertilizers or compost or chitin, it also contains a lot of total nitrogen, but it has to be mineralized first. And please keep in mind that this mineralization, it's also a microbial process. On the other hand, we also have biochar. Biochar may contain some total nitrogen, but this nitrogen is not mineralized at all. It's very stable enclosed within the biochar. So nitrogen mineralization, it's a microbial process, but in contrast also, the nitrogen immobilization, so the retention of mineral nitrogen by material, so it cannot be taken up by plants, it's also a microbial process. And we know that this risk for immobilization is higher for materials with a high CN ratio, like straw or wood materials. And we also see that the speed of this process is faster for materials, or the speed is higher for materials with a high decomposition rate. So sometimes for wood fiber, you can have nitrogen immobilization, but only in the long term and only at a very low speed, while for other materials, it can go very fast. And this brings us to the end of the cultivation, so the spent growing media. And also here, it means when you have less activity, you have a more stable material, and it also means more capacity for carbon storage. For instance, when you use it in the soil, so it can be like an option that you have spent growing media that you can reuse as biochar or as compost, and then in the end use in the field as a source of organic matter. And we tested it for different kinds of blends. So we had peat-based blends. We had blends with 40% peat replacement, and the same blend with biochar. We had blends with 100% peat replacement and the same blend with biochar. And we did stability assessments on these materials. And in short, we can conclude that they were all very stable. So it means that the carbon decomposition was very slow. So it means when you put these materials in the soil, 1 year later, you find back more than 90% of the original carbon. So these are very stable materials to be used in the field. So please also keep in mind that compost and biochar, they have different functions. In growing media, they also contain a lot of nutrients, mainly phosphorus and potassium, which are also plant available. So if you use compost and biochar, it can indicate that you can reduce the application of fertilizers. On the other hand, they also may contain a lot of inorganic carbon, and this allows you maybe for lime replacement. So if you have a blend with a lot of compost, it may not be necessary anymore to add additional lime, because this compost already allows you to increase the pH to the optimal level. So this brings me to the end of the presentation. So please keep in mind that new materials in growing media, they have a higher activity and a higher biomass. So it is important to figure out whether it is positive or negative. It is important to indicate or to know that nitrogen should be released, but it can also be immobilized in growing media. And all of these microbial processes can be inhibited by limiting factors. So for instance, if the moisture content or the oxygen content or the nitrogen level in your growing medium is not optimal, it can happen that your microbial processes that you expect, that they do not happen at the speed that you would like them to happen. So thank you for your interest, and we now move on.
Yes, Bert, thank you. We'll go straight to the next speaker. That's a colleague of Bert. So Jane de Bode is also a senior researcher at ILVO in Merelbeke, and she will tell more about the microbiome and the added value in the growing media. So Jane, the floor is yours.
Hello, do you hear me? Yes, okay, thank you. Today I will talk about the microbiome of growing medium, and first I will explain why it is important, the microbiome of growing medium. Secondly, I will focus on the techniques we used within the Horti-BlueC project to measure the microbiome of growing medium. And to explain each of the techniques, I selected a case study where we added chitin to the growing medium. And with this case study, I really want to demonstrate how these techniques can be useful and how these techniques learned us in the project the mode of action of chitin. And then I will make some conclusions about what we learned about chitin in growing medium. So first, the importance of a microbiome in growing media. So basically, we can divide the microbiome in growing medium in 3 groups. So first of all, you have the harmful microorganisms. So then we think about plant pathogens such as Phytophthora, or human pathogens such as Salmonella. Second, you have the beneficial ones. So they can be naturally present in the growing medium, or you can add them to the growing medium, for example, by using commercial products. Why are they beneficial? Because they have several functions. For example, they can promote the growth of the plant, or they can help the plant to defend himself to diseases. So they are help the plants to be, they help the growing medium to be disease suppressive. Then third, you have the saprophytics ones. So these are present in the growing medium, but they not really have a function, except when they are maybe with too many of them. For example, for fungi, if you have too many fungi in your growing medium can become hydrophobic, and then they cross the border to be harmful to the plants. Except for this this division in 3 groups, you can also look in 2 ways to the microbiome. First, you can look just at the bulk growing medium, or you can also look to the microbiomes that are in the rhizosphere of the plant. Of course, the microbes that are in the rhizosphere are influenced by what's present in the bulk, but the roots of the plant really select a specific microbiome, which is interaction is in interaction with the plants. So here you see an overview of the 4 techniques that we used in the Horti-BlueC project to measure the microbiome. And I will not go into detail, but the main message is that each technique measures something else. So it's not that you can replace one technique by another. So they are really complementary. And each technique measures something else, and it has advantages and disadvantages from to be very labor intensive, to be very difficult to do the data analysis. To explain the techniques more in detail, I selected a case study where we added chitin to the growing medium. And so we measured and we used these 4 techniques in this case study. We focused on 2 crops, on strawberry and lettuce. And for strawberry, we saw that disease suppression was correlated with an increase of microbial biomass as measured with PLFA. And we also saw that there was a shift in the rhizosphere microbiome, and this was measured with Metabar coding. For the lettuce, we saw that there was a plant growth promotion, and this was correlated with an increase in functional genes as measured with shotgun metagenomics. We also saw that there was an increase in plant growth promoting fungi, and this was demonstrating by isolating these fungi via plating. So as you can see in the Horti-BlueC project, we mainly focused on beneficial microorganisms that could be helpful in disease suppression and plant growth promotion. So now I go to the specific case of strawberry. In the strawberry experiments, we added 4 sources of chitin to the growing medium of strawberry. And here I explain which 4 sources it are. So we had the chitin extracted from crab shells. This was chemically extracted, and this was like a reference for commercially available chitin. Then we also extracted chitin chemically from shrimp shells. And third, we had chitin that was obtained by roasting shrimp shells at 200 degrees. And fourth, we had chitin obtained by roasting shrimp shells at 300 degrees. So after adding the chitin, we planted our strawberry plants. And after some weeks of growth, we inoculated the leaves with the pathogen Botrytis cinerea, and we evaluated the size of the infection on a score from 0 to 4. So on the picture here, you see the strawberry plants, and this is a score 1. At the end of the experiment, we measured the microbial biomass around the roots of the plants as measured with PLFA. What did we see when you look at the disease on the plants? As you can see, 3 of the 4 chitin treatments, we have less disease as compared to our control where we added no chitin. And remarkably, if then you look at the microbial biomass as measured with PLOV, you see that also in these 3 treatments, you have a significant increase in the microbial biomass as compared to the control where no chitin was added. For the chitin that was obtained from roasted shells at 300 degrees, we did not see a significant increase in the microbial biomass. If we then look at the Metabar coding results, so the Metabar coding of the rhizosphere of the strawberry plants, here you see the results of the fungal Metabar coding. And then when we first look at phylum level, I think you can really clearly see that again, the 3 treatments, there is a very clear increase in the purple group. And this, when we look at phylum level, are the Mortellella mycota. For the control where we did not add chitin, you see the purple group is very small, and also in the chitin that didn't work, this purple group is very small. When we go then back more into detail and we go up to genus level, you see that again here you see that there is one group that is significantly increased, and these are the group of the Mortellella. And this was not seen as spectacular in the control treatment or in the 300 degrees treatment. Okay, so now I go to the second crop we worked in. This was lettuce, and there we saw that the plant growth promotion was correlated with an increase in functional genes and in plant growth promoting fungi. And here, this is a very complex figure. For more details, I refer to the publication of Caroline, the tender. But I want to show with this figure is that with Metabar coding, we saw that 2 types of microbes increased. Microbes that have genes that are capable of the degradation of chitin, and microbes that are able to oxidize ammonium. And this is important because the chitin needs to be degraded and ammonium has to be oxidized so that the nitrogen can be released. And this release of nitrogen explains the plant growth promotion we see here due to the addition of chitin. Then as last part, because we also really wanted to demonstrate the plant growth promoting effect of these Mortellella. In the lettuce experiments, we clearly saw that there was also an increase in this Mortellella based on our Metabar coding, but we also saw visibly on the surface of our pots that in the beginning of the growth, we had like a white crust or fungal mycelium growing on the surface of our growing medium, but also out of the rhizosphere, we were able to pick up these fungi and we placed them on plates, as you can see here. And then we added spores to Arabidopsis plants who were grown in vitro. So here is a plate without these Mortellella spores, and here are plates with these Mortellella spores. And I think you can clearly see that if you add these spores, that the Arabidopsis grows better. That means that the fungi by themselves are plant growth promoting. So I want to conclude with some remarks, is that we saw that chitin stimulates the disease suppressiveness and the plant growth promoting fungi in the growing media, that the type of chitin, so the production methods, determines which microorganisms are stimulated, because we saw that only 3 out of the 4 types were able to stimulate these specific microorganisms or this Mortellella ceae. And then until now, with this microbiome analysis, in most of the cases we can only see a correlation between function and microbiome. So function can be disease suppressiveness or plant growth promotion, except for the very old technique of plating. If you plate these microorganisms and you re-inoculate it on the plants, you have a direct proof that your microorganisms have a beneficial function. In this case, it was shown with Arabidopsis and plant growth promotion. So thank you. And I also want to thank some people of the lab. So in this picture, you see the ladies of the microbiology group, but also all the partners and participants of the Horti-BlueC project. I want to thank them for the nice collaboration.
Thank you, Jane. And also Bart, to show your very interesting results that Ilvo obtained within Horti-BlueC, or some of the results, but because you did way more than you showed here. But thank you for presenting it so clearly. Now we're going to make the step towards using the blends in commercial settings. So a few other partners will present their findings on strawberry or tomato with the different blends that were provided by Jeroen from Aharis. And first we're going to start in the UK with Ewan Gage. Ewan Gage is the commercial trialist from ADAS. It's a research institute in the north of England, UK. So Ewan, I would like to give the word to you.
Good afternoon. Okay, so we've already had the goals of this project introduced as demonstrating the potential for alternative substrates for commercial production. Our contribution was really trying to help bridge the gap between laboratory work and what could be commercially meaningful to the wider industry. I'm going to talk about our focused work on tomato production. This is a very significant sector in the UK, which has largely transitioned towards the use of using coir as an alternative substrate, although some growers are still using Rockwool. There is desire to move away from these materials, particularly because of the length of the supply chain and the carbon footprint of using coir produced in Asia. So there was a lot of interest in the potential to produce homegrown alternatives to this. Aharis was capable of producing a significant quantity of the prototype substrates, and we looked at 3 substrates in particular: a plain wood fiber basis, a wood fiber based substrate amended with biochar, and a wood fiber based substrate amended with chitin. In order to demonstrate the commercial potential of these substrates, it was necessary to show that these materials had an equal, if not greater, performance to coir in terms of yield output and crop health and crop production. It was also necessary to start thinking about the commercial aspects of using these materials on the large scale, such as how much it would cost to fit out a hectare of a glasshouse with these composts compared to coir, and also how are they handled and used, and how does their stability and physical parameters impact their use on a commercial scale. As well as aiding growers in moving their growing onto our alternative substrates, it was also necessary to demonstrate some additional benefits of using materials if these were found. Biochar, for instance, there is already a strong interest in the UK sector for the use of the biochar to improve the disease resistance of a crop, either through increasing root health or through other processes boosting the disease resistance in the plants as a whole. This really fits in with the wider interest in terms of plant or root health management, but there is also an interest in optimizing nutrient inputs and the sustainability of production as a whole. Following some preliminary trials that were carried out in our laboratories in Cambridgeshire in 2019, we were able to demonstrate that these materials at least fulfilled the first of few of these objectives in demonstrating that equal, if not greater, performance could be achieved compared against commercial standards of coir and Rockwool blends when used for tomato production. We were also able to demonstrate that vegetative conditions of the plants were also improved, in particular with biochar. Given that biochar had strong performance, this was selected as the blend to take forward into the commercial trials. And so 2 full-scale commercial trials were carried out in 2020 at 2 separate tomato growing sites on the south coast of England. The first site used coir as their main substrate, while the second site used Rockwool as their main substrate. And so the biochar wood fiber substrate was compared against each of these over the full course of a production year. These were primarily scored for yield output to make sure the results are commercially meaningful, but we also took some focused experiments looking at the disease development that was observed under natural conditions in commercial settings as opposed to inoculations in the lab. Looking at yield output overall, when compared against coir, we were able to demonstrate that the marketable yield was comparable to coir production over the course of a year. This is really strong. This is a really strong piece of evidence to support that coir could be substituted for the wood fibre and biochar blend and still achieve a comparable level of performance overall. When compared against Rockwool, there was a small but potentially commercially interesting reduction in yield when compared against Rockwool. This is an interesting contrast to coir, but I feel this is more likely an artifact of the trials in that the Rockwool and biochar blends used in this experiment are likely to have had slightly different requirements in terms of irrigation and feed application. And because these slabs were being fed as if they were Rockwool, it's likely that they were being undermanaged and that this yield performance could be offset by improved crop management. We were also interested in looking at the development of root diseases. Crazy root is a significant commercial problem in this country for which there is no viable commercial control mechanism available. As we've already heard, biochar can significantly impact the microbial population and diversity of the substrate, and therefore we were hopeful that with cultivation under commercial settings, that biochar usage could be used to suppress the incidence of root mat symptoms in a commercial crop. In a trial that was carried out at the same site compared against commercial coir in 2021, plants were cultivated as normal and scored at the season end for the incidence of crazy root symptoms on a severity scale of 1 to 6. The data I presented on the bottom right-hand corner shows two control rows of coir with a biochar wood fibre row in the centre. The biochar row showed very little expression of root mat symptoms over the course of the season compared to significant root mat incidence seen in the neighbouring control rows, particularly on the left-hand side. When discussed with the grower, the feedback was very positive, that they felt there was a very strong impact on disease development, particularly in a crop that was prone to developing crazy roots. Taken as a whole, we feel that the commercial trials have been a success over the last 2 years, and we have demonstrated that the wood fibre blend amended with biochar can be used as a viable alternative to coir and Rockwool for commercial production. Feedback from growers commented on a variety of perspectives, looking at improved root health. There was feedback that there was greater penetration of roots through the trial substrate and less matting on the slab base, particularly in Rockwool. In terms of fruit quality, there was no significant differences when compared to either coir or Rockwool, with things like bricks coming back as comparable to the commercial control. So we're not impacting the fruit quality in any way. I presented some data here on the right-hand side with the biochar substrate compared with Rockwool. Slab water content was generally higher and much more stable for the controls, but it may be necessary to mitigate this to some extent, particularly if this has an impact on vegetative growth and the balance between vegetative and generative steering from the grower's perspective. Some of the feedback we received from the growers, that they felt it would be necessary to go through a process of optimizing the use of these substrates to ensure that agronomic needs are met. Particularly when the industry has got so used to using coir, there would be some translationary period into adopting these new materials and getting the best out of them in the commercial setting. They also felt that price was likely to be key. Concerns about sustainability, disease control and nutrient management, for example, will be secondary to the economic sides of production. So therefore it'd be necessary to make sure that price could be streamlined to ensure that it would be competitive with the existing commercial blends. Last but not least, some of the growers demonstrated that they were concerned about the handling of these new blends, particularly as they were quite heavy and bulky and needed extra labour to be put into place compared to Rockwool and coir, which can be put in as dried down light slabs. But again, this is something which is likely to be approachable from a systems approach in terms of manufacture, and could be optimized before wider scale commercial rollout carried out. The last piece of feedback that I had about this was that the grower that had been using these materials for the last 2 years has actively sought to use them longer term in his production because of the benefits of the success, benefits that he's seen from the trials and views them as a success, which I feel is really, really positive feedback about the output of this project. That's as much as I'd like to talk about. So thank you for listening. I'll now return you.
Thanks, Ewan. And indeed, that's very good news that growers are already trying to get more substrate towards them for using them in the long run. So very good news. Let's see if our colleague in Belgium has seen the same results or similar results with tomato. I would like to introduce Lotte Similon, a researcher here at the Proefcentrum Hoogstraat in Belgium. She's a researcher on tomato, and she did similar trials on a commercial scale as Ewan, and she will present her findings.
Thank you for listening to this little video. As you could see in this video, we compared especially the Horti-BlueC substrates with Rockwool because in Belgium we are used to grow tomatoes normally on Rockwool, that's the standard. And so for us it was interesting to compare the Horti-BlueC substrates with the normal peat substrates and the Rockwool substrates. I will tell you more about the difference between them during the growing season. During the growing season, we saw some differences in biological interferences, like in the slabs with plant fibers, we saw some fruiting bodies from a saprophyte. And we observed that during the planting period, because there during the planting period there was no drain in the slabs, and probably because of that, fruiting bodies from a saprophyte could evolve. But after 1 month within the growing season, there were no fruiting bodies anymore observed. That could be not good because the slab could be become hydrophobe, but after 1 month we couldn't see anything anymore. So and then during the season we also have to treat it with Sternemia felta to control scaridae, but that's something that's normal within organic blends to control that. Then some other differences. The slabs, the Horti-BlueC slabs were heavier than the Rockwool slabs, and that makes it harder for labor. Also, the slabs were more bumpy than Rockwool slabs. That makes it difficult to make planting holes, and that takes more time. the slabs, the Horti-BlueC slabs, were also more difficult to wet in winter, and so also to drain. But when we go in the summer, we observed a more stable water holding capacity and a more stable EC comparing with Rockwool. Like you already could see in the movie, production and fruit quality remain the same. So that were really good results.
Thank you, Lotte, for your presentation. Very good. So Lotte and Ewan used peat-free media blends to grow tomatoes on. We also tested different blends on strawberry, but these are peat-reduced, so not peat-free, but peat-reduced media blends. And Maarten Hofkes of Proefcentrum Hoogstraat in Belgium, a researcher on strawberry, a colleague of mine, he will present his results and his findings in the next presentation. So Maarten.
Thank you, Peter, for this nice introduction. So in the last 3 years of the project, we were able to finish 5 strawberry trials here at Proefcentrum Hoogstraat. So in total, we tested 20,000 of these new blends. So this is quite a large amount. And the blends that we have tested were blend 5, blend 6, 7, 8, and 10. And in all of our trials, we used the variety El Santa, which is a June-bearing variety. And with this variety, we did both single or 60-day croppings, but also double croppings to see the long-term effects of each new blend. As conclusion, we can say that in none of our different trials, we see a clear different effect towards yield or grading. And in these trials, we also monitored the drainage volume and EC, and regularly we sent samples to the lab for a nutritional analysis. And these results showed that these new blends did not bind or release any of extra nutrients. So this means that the fertigations or strategies, schemes or strategies that we give did not to be adapted when we are using these new blends. And in general, we can say that with using one of these new blends, we did not affect any side effects, so which is quite good. We did also a workshop where growers were able to visit the ongoing Horti-BlueC trials. And afterwards, we asked our participants to fill in a small survey. They have to give a score from 1 to 5 to each option. And a 1 means not important or not relevant. And a 5 means very important or very promising. The first question that we asked was which parameters are the most important to you for a more sustainable substrate? And then we see that the same production level, stable structure, and stability of the substrate is most important. So an equal performance of the substrate is asked. And this is also what we seen in the trials. So this is quite good. And a second question that we asked was which additives are the most promising, or do you prefer to replace the peat or the compost? And the options were biochar, wood fiber, and compost, and all 3 seems to be quite good for them or relevant for them. So it is useful information for us that growers are open for this more sustainable substrate. So that's the things I want to show and share with you today.
Thank you, Maarten. let's get back over to Ilvo. Laura Goldstein. She's actually coordinating all the presentations and putting them into the right order. So Laura has prepared a video where she will showcase the different Horti-BlueC websites, and she will give an explanation within the video how to use them and how to get all the information that from all the different research topics that we've did, and how to interpret them on the website.
Hello, let me show you the Horti-BlueC website. On the Horti-BlueC website, we are establishing an online collaborative platform. One of the features of that online platform is the decision tool. To get to the decision tool, go to the Horti-BlueC website and click Online Platform, and then Decision Tool. When you get to the Decision Tool page, click the image of the decision tool and it will open in a new tab. The decision tool shows you how a couple of residual materials can be processed or treated into new building blocks for sustainable growing media, energy, and CO2 fertilizer. The advantage of the tool is that you can access the information from different starting points. You can start, for example, from one of the residual biomasses and find out how it can be processed. But you can also start by clicking one of the processing methods or treatments. But you can also start on the right from one of the outputs that you want to obtain. To get more detailed information about the processing methods and the characteristics of the feedstocks, scroll down. If you have more questions or if you want to give feedback on the tool, click the contact button, write your email and your question or feedback and submit it. So I hope you will find the tool helpful and that you will enjoy trying it out. Another feature of the Horti-BlueC online collaborative platform is the mapping tool. The idea of the mapping tool is to map all stakeholders that are potentially involved in a valorization chain. So it can help to identify new connections within circular economy and to establish contacts between stakeholders to build a viable valorization chain. Currently, we have one map developed for shellfish contacts in the UK. This map can also be found on the Horti-BlueC website. Go to Online Platform and click Mapping Tool. Click on the image to open the mapping tool in a new tab. The map shows shellfish contacts in the UK. By clicking the symbols, you can get more information about the companies and organizations, such as contact details. For each of the valorization chains, Horti-BlueC has made a SWOT analysis. The SWOT analysis is actually the first step in illustrating the feasibility of the different valorization chains. A fact sheet with those SWOT analysis can be found on the Horti-BlueC website. Go to Project Outputs and then go to the valorization chain page. If you scroll down on that page, you can find PDFs with the SWOT analysis which you can download. So this is what the fact sheet for valorization chain 4 looks like. For each of the valorization chains, the fact sheets report the strengths, weaknesses, opportunities, and threats of one or more new solutions suggested by the Horti-BlueC project versus a current baseline scenario. So please go visit and discover our website and find out more about the valorization chains and the project in general.
Okay, so thank you everyone for attending and for asking questions. As a chair, I'm going to wrap up now, but I'm going to give the word back to Bart for a closing statement and his wrap up. So thank you everyone for attending and see you next time.
Okay, thank you, Peter. I would really like to thank you for your for chairing this session. You did a very good job. I would also like to thank Laura for the work behind the screens for making this webinar look so very professional. So thank you for that. Of course, also the presenters for their enthusiasm and the results they have shown. I would like to thank Hein Boon for his reflection, and of course the audience. You were fantastic. You raised a lot of questions, but it's not the end yet. So there are still some slides to go. So you will receive by mail a questionnaire based on this webinar. So we want just to know what you think about the results that we have presented. And if you have any remaining remarks or questions, you can also put them in the questionnaire. So please fill this questionnaire. It's important for us to know what is the impact of this webinar. So thank you already for filling this. If you want to know more about the project outcome, we are very proud to present the final Horti-BlueC newsletter. So you will receive the link also by mail, and it will also be available through this link in the presentation. So all presentations of today and also the recording and the fact sheet, they will be made available to you. So you can find there all the information. If you want to start playing yourself with the different building blocks that we have presented in the project, you can find more information as a video or as scientific papers, but also through the decision tool. So you can also use these links to find this information. And so I really want to thank you all for attending this webinar. If you want more information, you can find it here. So please also visit the Horti-BlueC YouTube channel with a lot of information, but also the website. And you can, of course, follow us on Twitter as well. So thank you very much and hope to see you soon back. Bye-bye.